the molecular biology lab in the other room. They have some projects that complement here the part of in vitro culture, and then they analyze genes, transcriptomes, they complement the study. There are also projects that include our lab, because sometimes in vitro culture, the goal of the in vitro culture is to eliminate some pathogens from the plants, so sometimes this work is made downstairs with us to confirm that the plants are free of viruses or whatever. We have some projects that together, each one with his speciality. Okay, we can visit here. You can interrupt me whenever you want. And have some This is not, I worked here some years ago. No, I'm not so close, but if you have some doubts, the person that works in each subject we can ask and to see what they can explain this. Okay. Laminar flux chambers. It's in asepsia culture, in vitro. This is some colleagues that work here with the multiplication of plants. They work with several species. We are going to see which are the main species that here is the
Can we take a photo?
Yeah, no problem. The work in the biotechnology lab is very time consuming because the part of the preparation of the culture's medium, the contamination that are always appearing, the sterilization of everything, so it's very time consuming and it's it's hard, it's a hard work. Virginia is a technician, and then we have Rita that is a PhD student. Okay? Here is where they prepare the culture medium. In the other room, they also have another where is the medium is the pH. Yeah, it's to prepare the medium, like here in big volumes. So this is where this part of the work. Here they store when they prepare the culture medium. According the medium is different according to the species or the objective, the goal of the work. We are going to see the chambers, and when you can see the plants that they are working on. So we have special medium for each species, special medium. The objective is the multiplication, or is the rooting, or is the somatic embryogenesis. According to the goal of the study, is they work with different culture mediums. Here, this flux, the air is filtered like this, so everything is sterilized. So you have to have too much care, otherwise you get contaminated everything and you have to start again. So that's the part not so friendly of the in vitro work. You can go here. The species that we work with, it's a little depend of the projects that are working, but there are some that are very typical from Alentejo region that are important in terms of, for instance, they work with olive oil. So there are some species, some olive varieties that give some characteristics to the oil. So typical from here, from this area. So they try to solve some problems that some of these varieties have, so they can produce and to multiplicate them. So one of the species that they work with is a variedade cultivar galega that is very typical traditionally from our oils, but the galega has a problem because usually the olives are propagated by rooting. So, but this specific cultivar, it's not easy to get roots. So they are studying the molecular mechanisms and trying to solve with culture mediums to propagate this specific variety. So this work is being the aspects of in vitro rooting of this cultivar and also from others is a work that is being performed here together with molecular biology lab, in which they are studying especially proteins and genes involved in the in vitro rooting. Another project that they are working with, it has to do with the somatic embryogenesis also in olive. It's the work that Rita is working with in her PhD. So if you want to know something more about it, we can talk with her. Then another project that is also that started with some years ago when I was here. You are working with it. It has to do with the propagation, in vitro propagation of rootstocks of walnut. This specific rootstock has some characteristics. I think it has to do with resistance to abiotic stress, water stress. So this project has been propagating. We are going to see the plants in vitro propagation of rootstock of walnut. This project that took place some years ago has an important this project intended, and I think they succeeded, to form at the end of the project a company, so they can produce. It's a type of project not only for investigation but also for transfer of knowledge. So they form the colleagues that work. By that time, they form a company, and they are already, that is here this is also, I think they have the name somewhere, I don't know. So they are producing walnut plants. So we are going to see here the in vitro chamber. Virginia.